Okay, so good morning to one and all, and good evening to whoever is from the other side. And thank you for the introduction and for having me here to talk about my recent paper on gut microbiome and aging. So, here we try to explore Asian octogenarians and their gut microbiome to see if there is any sort of potential gut microbiome signatures that we can identify, which can be used as a modulator for healthy aging. So, why is aging an important area to study? So, aging is something that really affects us all. It's not like a particular section of people who are going to get affected, but all of us across the board are going to be aging at one point or the other. And over the years, we have seen that the life expectancy has increased and there is a reduction in the rates of fertility, because of which there is a push towards more expenditure on healthcare. And we also see that this sort of aging phenomenon is specifically affecting a lot of Asia Pacific region, where we can see that the spending across the healthcare increases as is going to increase, is projected to increase as well because of the number of people who are aging. And we need to do something about this to try to see if we can identify any sorts of interventions that can promote healthy aging, because aging is going to be associated with its own set of morbidities and comorbidities that we are going to be subjected to. So in this phenomenon of global picture of aging, how does gut microbiome come into picture? So when we look at what happens to the overall physiology of the gut, we can see that with age there is a lot of change that happens, including an alteration in the ability to chew and swallow, and changes in the gut and the reduction in digestive enzymes, mucosal immunity, and so on and so forth, because of which there are going to be changes in gut microbiome. And this is not something new. People have been working on this for several years now. And there are studies previously which have shown that there are a few genes that are reduced with age and few of them that are increased with age. But more or less, these studies have been using 16S rRNA sequencing, although there have been a few studies in the recent years which are focusing on shotgun-metagenomics. And what we are trying to do in this study is basically to identify species level associations by doing a deep shotgun-metagenomic sequencing on the gut stool samples that we had obtained from the Asian or the Singaporean octogenarians. So here we leverage the SG90 cohorts. So this is basically a longevity population health study. So this was started way back in 1990s and it has been continuing. So it's a longitudinal study and it has been a part of other aging studies as well. And here we are looking at community dwelling participants who are generally considered healthy, that is, who are not in a nursing home or a hospital, but are generally living in a healthy environmental settings and are able to carry on their own work and are aged above 80 years old. So, since Singapore has predominantly a higher life expectancy, we are able to leverage this fact in getting our samples for the cohort. And we usually consider them as healthily aging, that is because they are not subject to any other a hospital or any other environment. So the data that we have are essentially from stool samples, where from which we get gut microbiome, and the physiological measurements, which include fasting blood glucose, triglycerides and cholesterol levels, inflammatory markers, and liver health indicators. And what we have in our data is basically from over 200 subjects of SC90. And in addition, with other cohorts which were previously published from our lab, that is a platinum metagenome work and a CPE based work, we leverage the controls from each of these studies to look at how the gut microbiome has an association with age. And we also used a publicly available data set, which is from T2D and the controls from there, to look at to combine the entire data to find out if there is any microbe associations that we can identify. And the demographics of our SC90 cohort is basically having, we have 234 subjects of multi-ethnic group, basically Chinese, Indian, and Malay, and we have about 95 male and 139 female. And for this analysis, for all the analysis, we considered only the Chinese ethnic group, because the other cohorts were from Chinese ethnic group, and we did not want ethnicity to be a confounding factor. And we got the taxonomic and the functional profiles from taxonomic classifiers, that's metaflan 2, human 2, and from a publicly available database, that's curated metagenomic data. And one point to note is that since these are from different studies, there are going to be inherent batch effects, and we corrected for batch effects using muffin and did a bunch of sanity checks to ensure that the associations that we find are robust enough to be reported. So starting with the initial set of result, we looked at the breakout distance and plotted them on a pico-A. And we saw that there was a progressive shift that we can see in the microbiome profiles, in that the old subjects usually tend to cluster on the top part of the quadrant compared to the younger subjects who were in the bottom. And when we looked at the breakout distances across the pico, PCOA, when we looked at the PCOA axis, we could see that there was a shift in the profiles that we can observe in the y equal to x axis, whereas this was not seen in the other axis, indicating that there is some sort of microbiome changes that happen along with age. And this was in combination with alpha diversity metrics. So we could see that as we age, the alpha diversity metrics, that's the richness, Simpson, and the Shannon diversity reduces, which indicated that there are changes in the microbiome that happens with age, which is dictated by loss of gut species. And this was actually an interesting observation, because the other studies from the Western cohorts had an opposite observation, where they saw that there was an increase in the gut species as we age, indicating that this could be an Asian specific signature that we are seeing. So next, looking at the species level association. So here we performed a GLM, that's using generalized linear model, accounting for all the other covariates. We saw that a bunch of species are depleted with age, and bunch of them are enriched with age. So looking at the depleted list, we saw that several species of Ruminococcus, which is Ruminococcus bromai and Ruminococcus obium, were depleted with age. And there were other classic examples which were also shown in the previous studies of depletion with age, that is Faecalibacterium protsnitzii, Ruminococcus, Rose R. inulinivorans, Roseburia inulinivorans, and Eubacterium rectale, that were depleted with age. And it was also well known that these organisms are classic butyrate producers. So butyrate is one of the important metabolites in the gut, about which I'll come to it in the next few sections. And when we look at the organisms that were enriched with age, we found E. coli, uh, P. dori, and a few Alistipes species, which are enriched with age. And it was also shown that Alistipes species are also butyrate producers. However, they differ in the way in which they produce butyrate. And butyrate, as I said, has a lot of implications on host health, because it's an energy for colonocytes, and it's also anti-inflammatory, and is often linked to gut-brain axis. Next, we looked at the correlation networks of the elderly and young, and we found that there is a stark difference in the network itself, in that the elderly network was pretty sparse compared to young, with a lower number of organisms acting as hubs compared to young, where there were a lot of organisms acting as hubs. And interestingly, the organisms which were depleted in the which were found to be depleted in elderly were found to act as a guild in both the elderly and the young group. And similarly, those which were enriched were found to be acting together as a guild, indicating that there could be some sort of a functional uh, role that all these organisms could together play, which we were interested in finding out what they were. So we looked at the functional profiles from the metagenomic data, and found that between different metabolic categories, first looking at the sugar metabolism, we saw that the pyruvate fermentation was higher in the younger age group, and the elderly age group was enriched with metabolism of simple sugars, because of the abundance in glycolysis that we found in the elderly extreme aging group. And when we looked at the vitamin and energy metabolism, we saw that particularly with respect to the synthesis of vitamin B, there was an enrichment in the elderly that was observed in comparison to the other age groups. And next, looking at the most interesting one, that is the amino acid metabolism, we saw that there are a lot of pathways within the amino acid metabolism that were enriched in the 61 to 70 age group. And one interesting thing that we found was that there was an uh, overrepresentation of lysine metabolism in the two old age groups. And when we look back at the literature, we saw that for the butyrate production, there were two specific or four different pathways that were involved. One of them is the classic Krebs cycle pathway, which uses pyruvate as the starting metabolite and goes till butyrate, and the other alternate substrate pathway, which uses either lysine, glutarate, or 4-amino butyrate. And since lysine was overrepresented, we were thinking if lysine is actually being used up for butyrate production. And to understand this better, we looked at the specific genes that were involved from the lysine to butyrate pathway and saw that the genes are particularly upregulate or uh, uh, genes are particularly higher in the elderly age group compared to the younger age group, indicating that lysine to butyrate could be an alternate pathway that is being used up in the elderly, perhaps as an expansion to the pyruvate to butyrate or the classic butyrate production pathway. And to elucidate this better, since these were from the shotgun metagenomic data, we wanted to look at whether it is also captured in aging mouse models. And for this, we leveraged one of the aging mouse model, which was recently established by one of our collaborators, where aging mouse were divided into two groups, one of them with healthy aging, another one with control. And the healthy aging group was essentially fed with alpha ketoglutarate, which is an endogenous metabolite which was shown to extend lifespan and compress morbidity. And what we found in this with respect to lysine to butyrate production was indeed that in the healthy aging mouse models, there was an upregulation of these, or rather an increase in these genes. I shouldn't be using upregulation because I'm not doing a meta transcriptomics here. So looking at the number of genes, we could see that there were much more significantly higher number of genes in the healthy aging group compared to control, indicating that the expansion could indeed be true, that there is a lysine to butyrate conversion that helps in aging mouse compared in addition to the other pathways that lead to butyrate production. So having done all this, we wanted to next look at specific associations with the clinical markers that we had. And in particular, we were interested in looking at vitamin B12 and inflammation because there's been a lot of studies going on in addition to the liver markers. So in the first case, we looked at aspartate aminotransferase, which is actually a marker of liver health. And very interestingly, we found that a positive association with Klebsiella pneumoniae, because Klebsiella pneumoniae is usually associated with liver abscesses, which is more commonly seen in Asians. Next, looking at high sensitivity CRP, which is related to inflammation, we saw that a bunch of previously reported organisms like Escherichia coli and Streptococcus salivarius, which were related to high sensitivity CRP. And there was another interesting candidate, Egertella unclassified, which was also positively associated with high sensitivity CRP levels. And next, we looked at vitamin B12 levels. So here we looked at the associations with the level of serum vitamin B12 with the gut microbiome, and we saw that several organisms such as Streptococcus parasanguinensis and Bacteroides coprocola were positively associated. And in literature, strains of Streptococcus have been shown to produce vitamin B12, indicating that these organisms could potentially be used in designing a non-invasive test to basically find out the levels of these clinical markers and associate with healthy aging. So to summarize my talk, we found age associated shifts in gut microbiome, which is associated with reduction in richness and a depletion of microbial guild with classic butyrate producing species in elderly, which was also compensated or expanded with enrichment in lysine utilizing bacteria. And we saw that there was a relative enrichment of vitamin synthesis pathway in elderly and association of specific species with serum vitamin B12 levels and other microbial associations with clinical parameters. So I would like to end the talk with acknowledgements to my mentor Niranjan Nagarajan and my first co-author Indrik Vijaya, who helped me with all the analysis, and all others who were involved in the study from my lab, as well as my collaborators who helped with the cohort data collection and the mouse model and made this a huge success. Thank you.